So in the beginning of mitosis, there is the first phase called interphase, which comes before prophase, and this is when two pieces of chromatin are found in the nucleus, and during interphase, the DNA replicates and we add another chromatin. And as we see here, this is what happens at the end of interphase. The DNA replicates, and this chromatin is made of DNA, and genes are found in this DNA. Interphase is done, we're going to begin mitosis. So mitosis is an important part of cell division because it allows us to make more cells. And making more cells is needed for growth, tissue repair, and development of your cells. So during prophase, the chromatin condense and form tightly packed chromosomes, which makes it easier for them to move around. So what we're going to do is crunch these up and coil them so they become smaller and easier to move around. And obviously this isn't very pretty. Normally it looks definitely better than this, but we're just gonna say that this is the coiled version of the chromosomes. Not very pretty at all. So in prophase, the chromatin coil up and condense to form a tightly packed chromosome. And this just makes it easier for them to move around. So I already did that. So this is what happens at the end of prophase. Important thing is that these chromosomes now have two halves, and each half is called a sister chromatoid. And sister chromatoids of chromosomes are completely identical. Another part of prophase is that in order to divide up the DNA, we have to move the chromosomes to opposite ends of the cells. So I just moved these chromosomes to the other end of the cells. And as you can see that there's a border right here. And so this is because there is a nucleus that these are trapped inside of, and at the end of prophase, this nucleus disintegrates and disappears. And they're still at other ends of this cell though. Now, the nucleus is gone, but it's still a part of a cell. And in metaphase, the chromosomes line up single file down the middle of the cell. So they're going to line up right down the middle of this cell like this. Now, in anaphase, these sister chromatoids are going to split up and go to opposite poles. So this means, that this half is going to go this way of this chromatoid, and this half is going to go this way. Same thing with this one. This one's going to go down here, and this one's going to go up here. And this is what happens in anaphase. Now, during anaphase, we need to make two new diploid daughter cells. And now the first thing that we need to do during anaphase is to form a nucleus around the new cells that we're making. So we have these two different chromatoids, and they both have new nucleus around them. The next thing we need to do is we need the chromosomes to uncoil so they can make chromatin again. So each of these chromosomes are going to get longer, which is hard to, show because it's just Play-Doh and it's not a real chromosome. But we're gonna make them longer, make them uncoil. It's not my best work, but all right, they're slightly longer, take my word for it. And now we're going to have cytokinesis usually occurs at the same time as telophase. So this means that the cell is going to get split in half down the middle. And this divides the membrane to make two new cells. So for our purposes here, I am just going to rip this cell in half. And now, these are a part of two independent cells. So, mitosis and cytokinesis are now completely complete. We have two diploid cells that are genetically identical.